Hello and welcome to the QMed webinar updates and trends from FDA, including recent ISO 10993 biocompatibility news, brought to you by Nelson Laboratories. My name is Brian Bunce, Editor-in-Chief for QMed, and I will be your moderator today. This webinar is designed to be interactive. Learn about today's speakers, view additional resources from our sponsors, share and talk about this webinar via various social media outlets, and submit questions by clicking on the widgets in the dock at the bottom of the console. Please ask questions anytime during the presentation by typing your question in the Ask a Question widget and clicking Submit. Submit questions as they come to mind, and our speaker will address them during the Q&A session as time permits. At the end of the webinar, we will ask you to complete a short survey. Please take a moment to fill this out as your feedback is important to us, and will provide valuable information on the subjects covered in this webinar, as well as how we can improve future broadcasts. I am pleased to welcome Allison Komiyama, who is a former FDA reviewer turned regulatory consultant who specializes in regulatory submissions, quality systems, and biocompatibility evaluation. She founded and is a principal consultant for Acknowledged Regulatory Strategies, which serves clients who develop and manufacture medical devices. Our second presenter, Thor Rollins, is a certified microbiologist and currently works as a biocompatibility consulting manager for Nelson Laboratories. He is a participating member in AMI 10993 ISO committees and plays an active role along with the FDA and regulatory committees in developing standards, discussing biocompatibility methods, and voting on changes to those standards. Allison, I now hand it off to you.
All right, thank you, Brian. And thanks to everyone attending today for inviting us into your offices to talk about updates and trends from FDA. Let's get started. All right, so as part of the Medical Device User Fee Amendments of 2012, which is also known as MDUFA 3, FDA agreed to meet certain goals intended to help get safe and effective medical devices to market more quickly. Among other things, FDA agreed to, 1, post a list each year of guidance documents that the agency intends to publish within 12 months of posting. This is also known as the A list. 2, to post a list of guidance documents that the agency aims to publish as resources permit. This is also known as the B list. And 3, update or delete old guidance documents, namely those that were issued 10, 20, and 30 years ago, to reflect the agency's current interpretation of or policy on a regulatory issue. FDA makes it clear that there may be resource constraints or new hot issues that develop over the course of a year that might keep CDRH from issuing every guidance document on the A and B list. The lists are updated each year, and it is good practice to review them as they provide insight into CDRH's current priorities for the fiscal year. The prioritized guidance documents that the agency intends to publish in fiscal year 2015 is shown on this slide. So this is FDA's A list. In the left column are the final guidance topics, and the draft guidance topics are in the right column. I apologize that the font is pretty small, but hopefully you can see them on your screen. We're nearing the end of the fiscal year, and you can see in black the guidance documents that have yet to be finalized or drafted. The topics in blue are those that have been published, and I've included their publication date. On this slide, I've included the B list. Again, FDA intends to publish these documents as resources permit. As you can see in the left-hand column, FDA has made a blanket goal to finalize any existing draft guidance documents. In the right-hand column, there are 7 topics that FDA hopes to come out with a draft guidance this fiscal year, and so far they've published 1. I know there is a lot of interest in the third topic on the list, the direct access genetic in vitro diagnostic testing, as well as the last topic, use of symbols and labeling. So I hope to see these draft documents published soon. The retrospective review documents that I've dubbed the C list can be found at the bottom of this link that I've provided here. FDA invites anyone who's interested to submit comments on any of the topics presented on these 3 lists. You can submit draft language, suggest new or different guidance documents, push to prioritize specific guidance documents, or suggest that CDRH revise or withdraw a final guidance document that was issued previously as part of the retrospective review. So today I would like to address just 3 of these guidance topics that FDA has published this year from the A list. I've outlined them here in red, and I will cover them in this order. Since many of you are on the line to hear Thor's presentation about recent updates on the ISO 10993 guidance document, and I include myself in that group, I will keep my portion relatively brief, and I'm happy to field any questions at the end of the presentation or via email. The first guidance document is titled Intent to Exempt Certain Class 2 and Class 1 Reserved Medical Devices from Pre-Market Notification Requirements. It was published on July 1st. However, a newer version was just issued on August 14th that includes some additional exemptions and language in the scope of the document. For each guidance I talk about today, I've included the link to the document at the bottom of each title slide for easy reference. Sorry, I'm having a, there we go. During MDUFA 3 negotiations that I mentioned earlier, FDA also committed to identifying low-risk medical devices to exempt from the 510K pre-market notification requirements. FDA has identified specific unclassified medical devices, Class 1 reserved medical devices, and Class 2 medical devices, for which FDA believes pre-market notification is not necessary to assure safety and effectiveness. However, the scope of the guidance makes it very clear that it does not intend to exempt the devices listed in the document from other requirements such as registration and listing, labeling, good manufacturing practices, and medical device reporting requirements. These are often referred to as general controls. In total, FDA exempted 117 product codes under 11 different device types in this guidance document. I've listed 3 examples here of some of the device types that have exemptions, including cardio devices, dental devices, and ENT devices. I reviewed some of the 510K summaries for the devices cleared under these regulations, and many of them included performance testing, biocompatibility, and clinical data to demonstrate equivalence to predicate devices. With the publication of this guidance document, FDA no longer expects these medical device manufacturers to submit a 510K. While this document is useful to those wanting to get their new device on the market, it is also valuable to those already marketing their device using these product codes. For example, perhaps your company wants to make a design change that in the past would have meant a new 510K. Now you can make your changes and remain compliant as long as you follow the requirements of the general controls outlined in the previous slide. So for each guidance document I'm covering, I've included a Thanks FDA slide to give you an idea on why the publication of this document is beneficial to industry. For this guidance, it's pretty clear that these exemptions allow many medical device manufacturers to save precious time and money when getting their medical device to market. I believe it is very likely that FDA will continue to exempt more devices in the years to come as the agency becomes more familiar with certain devices that pose a low level of risk. The next guidance I would like to cover is titled Reprocessing Medical Devices in Healthcare Settings, Validation Methods, and Labeling. When the draft of this document was released in 2011, it was actually quite controversial as industry claimed FDA was stepping outside its jurisdiction and was trying to regulate the practice of medicine. However, consumers were equally up in arms after infections caused by dirty instrumentation were leading to some serious adverse events in patients. During a meeting at FDA in 2011, one presenter demonstrated that bone, tissue, blood, and brush bristles were left inside medical devices after the cleaning. So actually, when this guidance was finalized this past March, it was just days after the news broke about that superbug outbreak linked to the use of contaminated endoscopes and that had undergone cleaning protocols before they were reused. I believe this guidance document is critical in helping make pre-processed devices safer for patients, and I have definitely noticed an uptick in questions from FDA regarding cleaning validation of instruments. FDA makes clear that while reusable devices have become more complex during recent years, there are significant advances in the technology of reprocessing devices to help mitigate the risk. Manufacturers should also design their reusable devices with an eye for effective cleaning. The 4 reprocessing situations listed here are covered in the guidance document. They include reusable medical devices that are either supplied sterile or non-sterile and are processed or reprocessed prior to use. Reprocessed devices that are reused only by a single patient, or single-use devices that are supplied non-sterile and require reprocessing or processing before use. The document gives manufacturers a decent overview on how to come up with a successful reprocessing plan. This includes point-of-use processing to prevent drying of contaminants on the device, thorough cleaning, followed by disinfection or sterilization. Appendix B in the guidance is a great resource to understand these steps in more detail. Overall, I believe this document is helpful for manufacturers as it offers resources for developing your reprocessing instructions. It outlines 6 key considerations for your labeling, gives guidance on how to pick your worst-case device or protocol to test, and it also clarifies the documentation FDA is going to expect in your 510K, PMA, HDE, de novo, or IDE submission. Finally, I found a few of the appendices very helpful as they clarify common questions industry has with regard to reprocessing definitions and sterilization cycles. Finally, I thought I would touch on the draft guidance about direct part marking, as I've been fielding a lot of questions lately with regard to unique device identification, which is also known as UDI. The UDI rule published in 2013 established a unique device identification system for medical devices. It requires that all labels bear a UDI and that specific information be submitted to FDA's Global Unique Device Identification Database, which is also known as the Good ID. Part of the regulation stipulates that certain devices bear a UDI direct marking, namely those that are to be used more than once and are to be reprocessed before each use. Compliance dates for UDI vary depending on your device class and can be found at the link on the bottom of the slide. So what is direct marking? It means that you affix the UDI permanently to the device itself through etching, attaching a permanent plaque, or affixing a permanent tag such as an RFID to the device. It is FDA's belief that because these devices are intended to be reprocessed and reused, it is likely they will be separated from the original labels and packaging. Direct marking will help ensure identification of these devices throughout their life cycle. So what devices need direct marking? It is really important you review this document before you move forward and start etching the UDI all over your device. It really is intended only for devices that will be used more than once and that will be reprocessed before each use. So I've included FDA's definition of both these requirements here, as well as their understanding of the word reprocessing. In addition to explaining what devices will need direct marking, the guidance does a good job, I think, outlining exceptions to the rule. For example, if any type of direct marking would interfere with the safety or effectiveness of your 510K cleared device, FDA states you would qualify for an exception and would not need to comply. And I apologize, number 2 here is a typo. It should read that guidance clarifies what FDA will expect with regard to submission. For example, if the direct marking will require a new 510K or supplemental PMA. I'll add here that this document is still in draft format, so you can expect to see this guidance finalized as we approach the FDA compliance date. So I chose these 3 guidance documents on the A list today because they are all somewhat tied to the draft ISO 10993 document that will be finalized soon. For example, if you're a newly exempted device and no longer need to demonstrate equivalence to a predicate device, you still need to consider whether your device should undergo any biocompatibility assessment based on its patient contact. Similarly, you might consider doing a biocompatibility evaluation on your device if it will require direct part marking but your concerned safety or effectiveness might be compromised. So theyre will now talk about how FDA is evolving its views on biocompatibility and how it is taking more of a risk-based approach to understanding the potential material hazards associated with medical devices.
Thanks, Allison. And the future of biocompatibility is something that I really enjoy talking about. And if you've been in part of my previous webinars or seminars, you know that is a changing target. I started in this industry about 14 years ago, and the view of biocompatibility has really changed in those 14 years, up and down all over the place. But where we are now is actually a very good scientific foundation, and where the future is looking is also very promising as far as what is biocompatibility. So as far as the past approach goes, I think everyone is pretty aware of this chart. And so for those of you that maybe have never seen this chart before, which I find surprising, but if you haven't, I'll explain a little bit. This is in the appendix of ISO 10993-1. Before the latest update of 10993-1, this was actually in the body of the standard. And this has always been kind of the approach that medical device manufacturers take to biocompatibility. And so the chart is meant to guide you of what testing endpoints or what biological endpoints are necessary based on the contact type of your device and the duration of that contact. So the more severe contact a device has, or the longer contact that device has, the more testing endpoints should be addressed. Now, this isn't really going away necessarily, but in the previous approach to this chart, manufacturer would just look at their device, they would evaluate the contact, and then they would say, okay, so these are the tests I need to do, and I hate to use this word nowadays, but this is what it was a checkbox mentality. It was, okay, so there's my test, check, 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 and they would do that suite of testing. They would submit it to their regulatory authority, whatever regulatory authority they were looking to submit to. And then they would sit back and field questions if there was any questions, but usually it had to do with maybe some test-specific questions. But you pretty much passed into the regulatory arena with checkboxing those tests. Now, the downfall to this approach, and maybe I guess it's your point of view, maybe it's the benefit or the downfall of this approach, is you didn't really have to understand a whole lot about your material and its potential risks. After all, that's what you were doing with the testing, was trying to address those risks. And so you would just submit the material. You didn't have to do a lot of homework from your suppliers or get a lot of information before from their suppliers unless you were just doing it for due diligence. And then you would submit it for the testing. The testing labs would do those tests and give you a report back and say non-sensitizer or non-irritant, and you'd at that point feel confident about your product and its impact in the body. The other thing that also was hard is you didn't have to understand anything about the testing itself. You would hear words like sensitization or big long words like maximization or local lymph node assay or magnetism Caligman and things like that, and you could kind of just take those in and repeat them without even understanding exactly what they meant. So for some people that might be seen as a benefit, but reality it's a disadvantage because there's a lot of time and money spent into the design of these products to get to one of the most lengthy and costly preclinical evaluation parts of a new device, the biocompatibility, and not having the same kind of attention given to those endpoints. So to kind of give you a real-life example, I guess a parable, this is my car example, and it's kind of real life for me. So on the left there, we have a 1967 Mustang. That was my very first car. And on the right is a Ford Focus, my car I drive now, not the RS version. I wish it was that car. It's not that one, but let's pretend. So the difference between the past approach for biocompatibility and the new approach can be a parallel to these cars. They both are functional. They both get you to where you need to go. But in reality, the one car gets you much more efficiently. Trust me, I know my gas mileage in that 67 Mustang. So it gets you there much more efficiently, and it gets you there more safely than it does in the old approach. we still have tons of medical device manufacturers that are comfortable in their old cars, that are looking at just doing the checkbox mentality. But the FDA is progressing beyond that. They are going to the newer approach, which makes some people uncomfortable. It's kind of like when you open up a new car engine compared to the engine of the 67 Mustang, and you see how complex or different it may be. And even though the principles are the same, it seems harder to do. And so they tend to just go back to the simple car, with not taking advantage the efficiencies and the safety that goes into the new approach. And so for that reason, I'm here to kind of talk about the new approach, what is going to happen, what has been happening, and what the new standard that's going to, or the new draft document that's going to go up, we expect to kind of have in it as far as what we how to approach biocompatibility. So the future approach really has three kind of things you have to think of. First, you have to think of a biological or toxicological risk assessment for the patient's safety. We have to start evaluating the materials that we're putting in these medical devices. We have to look at their history of use. We have to look at the processing that goes into those materials, what residuals may be there from their processing, and what impact do those materials and processes potentially have on the body. We also have to do our material and chemical characterization, and those are different. A lot of times I think we use those interchangeably, but a material characterization incorporates much more than just the chemistry of that device, and we'll kind of touch about that in a minute. And then only after you've done your toxicological risk assessment and your material or chemical evaluation, then you can do biocompatibility testing where needed or if needed. And that really comes down to the endpoint or the results of that initial evaluation and material and chemical, and the assessment done on those endpoints as far as what drives the testing or what testing is needed. So the definition of risk, and for me this is the ultimate form of biocompatibility, right? We do biocompatibility to evaluate risk. And the checkbox approach historically is what you did to evaluate risk. So ISO 14971 defines risk as a combination of the probability of occurrence of the harm and the severity of that harm. And so to translate that into maybe normal language instead of standard language, that picture I think that there says 1,000 words. We have the cheese, right? We have a safe functional device that we want to put on the market. But any material that's foreign to our body has a potential for adverse risk to the body. So we have to understand that risk and the benefit of the device, and we have to weigh those two, and then take precautions and maybe look at how we can limit the potential risk of that device to increase the benefit. And that's really what biocompatibility, the new form of biocompatibility is we want to understand the risk that's associated with the materials and the residuals of a device, then look at the benefit that the device has in the body, in the patient, and then make a conclusion, and then any gaps in that risk assessment we fill in with testing, with our historical testing route. So this is the flow chart out of ISO 10993, the current version of 10993, and the flow chart kind of helps us go through a decision on where biocompatibility is needed. And I know it sounds elementary, but I always talk about that first diamond, the first decision point, which is, does it have patient contact, direct or indirect patient contact? I see a lot of evaluations being performed on portions of the medical device or on materials or things in a hospital room that do not have patient contact. And if that's the case, then ISO 10993 does not comply. I'm not saying that you still may not do a risk assessment internally as far as safety goes and evaluate failure modes and worst case, but to go through a full biocompatibility test system on these types of materials are just not necessary. And when we're looking at limiting animal testing when involved with biocompatibility, this is one of the things that I think we should look at first, is what is essential to test as far as impact to the patients and what is more of a feel-good and how we can address that without doing unnecessary animal testing. But the other point that I want to bring out on the flow chart is that after you've made that decision of patient contact and you know it's something that has risks to the patient, Then we go through a series of material questions. We talk about history of material. We talk about predicate devices. We talk about other end questions that we have to address when we're looking at the materials. And a lot of times we don't follow this flowchart. Like I mentioned before, we just jump right to the testing. And if you look at this flowchart, the testing is the second to the last down at the very bottom is when we actually get to testing. And there's a lot of other questions and endpoints that may happen after we get or before we have to get to the testing box. So that's what we're going to kind of talk about here. Our first step of a medical device is material evaluation. And I know the engineers out there, they go through a lot of a material decision when they're looking at making a device. But most of those decisions happen to do with the functionality of the device or the cost of the material or how easy it is to get that material and these kind of questions. But a lot of times they do not look at the biological impact of those materials. And really, you want to find a material that has the information that you need to make a safe evaluation. And it may save you a lot of money and time, specifically time, on the back end if you can make a wise choice on your materials when you're looking at biocompatibility. So, what are the challenges to this approach? Kind of like the brand new car, when you open it up and you look at that engine and it's complicated and you have to kind of understand things a little bit more. Sometimes I think this is what these challenges are what scares people away from really embracing what we want the biocompatibility assessments to be. So the first thing we have to do is understand your product. And once again, this is not necessarily from how it works. I think most engineers have a pretty good idea of how a product works, but more on how it impacts the body, understanding the product or the risk associated to the end user of the materials and processing that you have on your medical device. Then you have to understand the changes that may happen to that product through its lifestyle or life cycle. So one of the things I always talk about is accelerated aging or sterilization. What impact does aging on a device have? Maybe nothing on certain materials. Other materials may age poorly. Or sterilization. Radiation may have a big impact on certain materials. So a material may be safe, may have plenty of biological data done on the raw material, but that's before any kind of sterilization has been done. And so we need to evaluate what impact sterilization may have or repeat sterilizations may have on a certain material. Then we have to understand the impact of leachable substances within your product. And for me, this is one of the top keys to fully understanding the risk of our product to a patient. Chemical characterization has been getting a lot of buzz lately, and it is one aspect of material characterization to try to understand the leachables that come off of your device. The reason why I think this is so important is because for the last 30, 40 years, we've been relying on animal results to show safety. But in reality, we don't know exactly how safe those medical devices or how safe those tests are in predicting the clinical impact of a device. We have a history of safe medical devices using those tests, but now that we're using chemistry and we can look at published data to basically come up with a risk assessment on each compound that comes off a device or mixtures that come off a device, now we're starting to reevaluate the sensitivity of these animal tests or these other in vitro tests. Are they as good a predictors as we originally thought? We're just barely scratching the surface of comparing the results of these material and chemical evaluations to the history of animal tests. And my assumption is that we might come up with better approaches, more sensitive and better predictors of safety by looking at the chemistry and material of devices than we would when we just inject into a guinea pig and sit back and watch what happens. So that gets us to the next point is we can predict clinical safety from only a few tests depending on the material and on the intended use. Those kind of things can have a big impact on biocompatibility. Now, once again, this may change over the lifestyle of the product. And so even though the rule of thumb is that most of the leachables will come off a device right away after it's been processed, the longer something sits around, the more volatiles might come off the device, and to limit the amount of extractables, leachables, or reduce the amount, still degradation may happen over materials, certain materials over time, and we have to keep that in mind when we're looking at safety evaluations. So, understanding the product, we have to look at the raw materials that's in your product, the consistency, the history of that material. We have to look at the patient contact of your device. What kind of impact could it have based off of the device impact with the body? Geometry can play a big role, only for specific outcomes though. In particular, when we look at what testing or biological outcomes are most impacted by geometry, the ones that come to mind for me quickly are hemocompatibility. Hemolysis actually has a big impact, or thrombogenicity and other hemocompatibility endpoints with the geometry of the device. Sharp edges can cause hemolysis. Rough edges can cause hemolysis. Occlusion of arteries or veins can cause thrombo. So these are the things that we have to look at for geometry when we look at biocompatibility. Also, implantation geometry can play a role, how the tissue ingrows into the material and what kind of response that ingrowth may have compared to a controlled material that we're using. So once again, we have to look at the geometry. Even though most of the biocompatibility tests we run are extraction tests that look at liquid that extract, you might not think that geometry plays a huge role, but in some endpoints, geometry can play a huge role. Then also we have to look at failure modes of medical devices. If you're using a balloon, for example, which is the one that comes to mind in the first, if that balloon bursts during use, what new chemicals or compounds are exposed to the body, which may not have been intended through the use of the device without that failure. So those are some of the endpoints or questions we also have to ask. Next, we have to do a toxicological safety assessment. And really what this means is we want to look at the device. We want to look at the exposure and what chemicals are coming off and what compounds and the mixtures, and then look at published data and make an assessment. Now, in truth, most medical devices are made out of materials that are safe. And we see a million times over and over again, I get that statement all the time. Why do I have to test this device? It's the same material that my competitors are using. It's the same materials that we've been using forever. And if that's the case, that's great. That's stuff we want to use in our assessment, our biocompatibility risk assessment. But at the same time, what are you doing to that material that your predicate device or your competitor device isn't doing? What colors are in there? What colorants? That's a huge question if you're using colorants. What residual detergents or oils or machining fluids may be on there that may impact biocompatibility. So that's where the chemical assessment comes into. We really get to see what comes off your device and make an assessment on the risk associated to the body based off of those compounds that come off the device. So the difference between material and chemical characterization. So this is the first step of biocompatibility. I showed you the flowchart, which is material and or chemical characterization. Material characterization goes into much more than just the chemistry. It goes into the history of the materials. It goes into the geometry, like I mentioned, the sterilization procedures, where it's being used, other endpoints that we examine. The chemical characterization, where we look at the leachables and extractables off a device, are just one endpoint of that overall evaluation of material. Sometimes when we do a material evaluation and we look at how the device is used, if it's on skin, for example, or very limited or low risk use, then a chemical characterization may not be needed. We might be able to evaluate risks for the patient just based off of the material evaluation. In other aspects, the material evaluation may tell us that we need a chemical evaluation to address some of those risks. And then depending on the chemistry, we may need to address other risks by actually doing biocompatibility testing. So the material characterization involves a review of the materials at the manufacturer and not just the final product. So we go through all the materials in the manufacturing process and review the potential impact into the body. Next, we have to have good communication with our suppliers, because now that we're basing it off those materials, if we have a change in those materials, we need to reevaluate the impact of biocompatibility. If we have a change in the processing of those raw materials, we need to reevaluate biocompatibility. So great communication with the supplier is essential. And honestly, this is one of the biggest headaches that we get when we do an evaluation for risk, is when we try to work with suppliers. Some suppliers understand this very well and are very willing to work with us on a material evaluation. Other suppliers seem to put up a wall and think that maybe we're trying to steal all their secrets on how to produce that material. And so they are more difficult. And something like this can have a big impact on how easily and quickly we can perform a material evaluation. Good communication with suppliers is essential. So what do we need for manufacturers in that relationship? We need to have manufacturing agreements in place, composition disclosures. We don't need to know how to reproduce this material, but we do need to know what goes into the manufacturing of these materials and what's in that material to make an assessment on risk. Like I talked about processing aids and residual chemical disclosure. MSDSs are very useful, but sometimes do not include everything that we need to have on evaluation, so sometimes we need more than that. And then obviously it's great if that material has a device master file with the FDA, but that being said, remember that device master file is probably on the raw material only, so even though we can use that as a great cog in that wheel, we still need to address what you're doing to that material. It's not enough by itself just to have a device master file with any regulatory authority. The next step is that biocompatibility assessment, and this kind of pulls it all together, your material assessment, your chemical assessment if needed, your testing if needed, and kind of comes up to how your biocompatibility is assessed. What risk is it really associated to the patient, and have you addressed all those risks? Once again, Table A, this is right out of the standard. Table A1, the one I showed you at the very beginning, is not a checkbox approach. And we are going to try to, in future revisions of ISO 10993-1, it looks like we're going to actually strengthen that approach, that we really need to evaluate each one of those endpoints and look at how we address those and not necessarily just by testing it. Once again, the difficult part of this is it does take experts in different fields to be able to come up with the right decision. So you have to have experts that you trust to be able to make those types of assessments and correct decisions. But if you do have those experts and you are able to make those endpoints successfully, you can save yourself a lot of time, especially time, in your biocompatibility evaluation. And the last one right there on that slide is chemical characterization is an integral part of biocompatibility assessments and evaluation. I do want to put a little caveat there for certain devices. I talked about low-risk devices that have minimal contact with the body or certain materials. These may not need a chemical evaluation or a full-blown leachable extractable profile to come up with a successful evaluation. But for certain devices that have more aggressive type of contact, the chemical characterization is an essential part of a biocompatibility assessment. So to kind of sum up this beginning part, testing is not mandatory under 10993-1. It's just one way to kind of address risk associated with your device, but there are other ways to do so. That testing is determined by the risk of the device to cause the patient harm, and we want to kind of address those risks through it. And these are some of the ways that we can do testing, quote unquote. So we can do in vivo, and that's the animal tests. We obviously want to try to minimize that as much as possible, not just because they're usually the most expensive and long tests, but because of the animal impact. We really want to try to do other approaches first. For example, in vitro evaluations. Can we be more sensitive with in vitros? We are investigating new in vitro tests for irritation and sensitization that both look very promising, and initial results do seem to be they may be a little bit more sensitive than the animal models. So they may be able to be used as a screening test before jumping right into the animal tests. We're working on round robins, specifically on the in vitro irritation right now in the ISO group, to be able to try to add that test to the standard. Clinical experience, history of safe use, and the material assessments can all be done as testing, dry lab testing, so to speak, to evaluate biocompatibility. Now, to give you an update in the last 5 minutes here, most of you should be familiar with this draft Allison talked about. This is the draft that came out in April of 2013, May, I guess, as the official date on there. And this is the new draft, or to replace the G95 draft for biocompatibility. I've given previous webinars about what's in here, but we expect this to be finalized this year. And honestly, from what I understand, there's going to be quite a few differences between this draft and the new one. It's going to probably incorporate a lot of discussion that we recently had in the ISO document or ISO committees with the FDA involvement. And it's going to be hopefully leaning more and more on scientific assessment of a medical device and not just a checkbox, which is in this current draft, but it will be emphasized more. Now, this is the link for the draft document in case you haven't read it yet. So in previous webinars, I've broken this down into three categories for the draft. Summary of ideas. These are things we've already seen from the FDA. They're just now written down, which is great for references. They actually give us, once again, kind of borrowing from Allison, thanks FDA, this is great. They've been giving us justification ideas or information we need to justify out of testing, and then things that we may be impactful to us going forward. And so I'm just going to go through these on the sake of time pretty quickly. The first one is justification. We talked about weighing the benefit of device to any potential risk associated with that device. So, once again, all these devices are hopefully meant to improve the quality of life. And ultimately, I think every single one of us, that's the reason we're in here. We're in this business to make life better for the moms and dads and children out there. And so each risk should be outweighed or weighed against the potential risk of that or benefit of that device. Now, it says in there, reviewers are encouraged to apply scientific judgment when weighing alternative data for animal testing. And in my thought, that's just not reviewers, but I think medical device manufacturers should use scientific judgment when looking at approaching biocompatibility. You should apply scientific principles on addressing these risks, and then knowing when it's in your best interest to evaluate those risks by using testing or other approaches which we talked about. Now, literature can be used, and these are all kind of quotes right out of the draft. Literatures can be used to justify out-of-systemic toxicity tests, but correct data should be used in the corresponding endpoint. So once again, if we're looking at literature at the compounds to address toxicity, well, we want to look at, one, the right route of exposure, so make sure the literature has the similar route of exposure, and if not, we have to do certain correction factors. We also want to look at the right endpoint as far as time goes. Was it an acute test? Was it a subacute? Was it a chronic? We want to make sure those right endpoints. And we also want to make sure that the testing itself was done scientifically and it's a peer-reviewed kind of test. So all these things can be used, but if we can find the right literature, we can use that as kind of a key to unlock the risk for systemic toxicity. Other justification ideas. Chemical characterization or biocompatibility testing can be a supplement to the biocompatibility overall assessment and can be used to minimize animal testing. Once again, we really want to try to minimize that animal test unnecessarily. It has its place, and when we need to address risk through the animal testing, we can. Now, this is something that helps you understand this next point, what the FDA thinks about when you're looking at a predicate device on the market to say that you don't need to do any testing or limited testing. And once again, just because your predicate device or your competitor device has the same material as your device doesn't mean that you can just say, oh, it's the same as this device, I don't have to worry about it. These are things that you have to address: the intended use, the physical form, formulation, processing component, which is one that I think is key. Storage conditions are the same. Then you can start looking at limiting your tests. So these are some of the endpoints you have to address in your overall assessment before you look at addressing it through testing. This next one is the one that I wish more people use, which is functionality testing to replace some of the biocompatibility. This is specifically with hemocompatibility, but all other endpoints can be used also. Lots of times these devices are put in functionality tests to make sure that they're working, and they use animals that are in clinically relevant conditions, in clinically relevant positions in the body. They might have weight on them if they're weight bearing, and so these are good predictors for biocompatibility, and we can use the evidence if the test is designed correctly, which work with your testing lab. Even if you're not doing your functionality test in a CRO, most CROs should be able to help you set up a testing protocol to be able to address some of these biocompatibility issues. And these things can help you get out of biocompatibility testing. Now, some of the things that may impact us going forward. The first one kind of talks about multiple contact with the body. If your device has multiple contact, for example, a delivery device compared to the implant that it delivers, A long time ago, before the draft, we've always had people who try to save money by combining those together in the same test. Because they have different contact types and because the surface area differences can bias the tests, you really have to test those individually or assess those individually. Once again, we're not necessarily testing them, but we're assessing them. But those assessments should be done individually. The other thing that we're interested to learn a little bit more about in this final has to do with the LAL test. The LAL test is a pyrogen test that looks at endotoxin. And in the new draft, it looks like the FDA may be requiring LAL tests for not just things that have lymphatic or cerebral fluid or circulatory contact, but anything that's labeled as an implant. So that would be orthopedic potentially. And we're looking at to see if that is also true. And then the other one I want to talk about as far as possible impacts is that bottom one, which is the new chart. And this is the chart right out of ISO, the new draft, and that's why it's a little, numbers down there are a little messy. I apologize, but this is right out of the draft. And if you can read it down there, we have circles, and those circles have always been possible tests that the FDA specifically, this is off the G95, may consider necessary, but were really looked at as testing that nobody did unless they had materials that the FDA were worried about. Well, the new draft has out there that these evaluations should either be included in submission, either by inclusion of the testing or a rationale for its omission. So now these circles have become more important. And in fact, we may see the draft, the new finalized document, even have more emphasis on this, where the testing itself should be addressed no matter what. Each one of these endpoints should be addressed in your evaluation. And this is once again putting more emphasis on that material and chemical evaluation, where we need to know our materials, we need to know our devices, we need to know the risk that is associated to the patient, and then address those with scientific information, either through material evaluation or chemical evaluation. And then only at that point, if we have gaps in our assessments, do we go to that testing to answer each one of these endpoints. And so the FDA is pushing more and more away from just checkboxing and requiring individuals to be thoughtful and have a scientific approach to performing biocompatibility. So thank you for your time. I think now we will leave the last little bit for questions and Q&A.
Thank you, Thor. If you have a question, please submit it now using the Ask Question box in the lower right-hand corner of your screen. If we're not able to answer all the questions during the webcast, we will share the remaining questions with our speaker, and he or she will respond to you directly. So our first question is, if a device touches the skin for 5 to 10 minutes, it falls under the category of contact time of being less than 24 hours. In such a scenario, do we need to perform biocompatibility testing? I'll turn that one over to Thor.
Yeah, that's a great question. It's kind of what I was mentioning before, where testing in the quote unquote, right? I mean, we want to evaluate the risk. So skin contact can still be a risk. So it really depends on the materials and the processing potential residuals on that device. Obviously, something that has skin contact is less of a risk than something that's implanted in the blood. And so the evaluation may conclude that some of the testing may not be necessary. And that depends on what endpoints we're looking at, what the material is made out of, what processing is done to it, what's the sterilization of that, and then all those kind of questions that are specific to that device then can lead you to that analysis, that biocompatibility assessment. And depending on the outcome of that biocompatibility assessment, limited testing may be appropriate. It just depends on the initial assessment.
One more quick question. When will new in vitro methods be added to ISO standard, or to the ISO standard?
That is a great question. If I had you sitting here, I'd give you $5. But that question is kind of varied, although we are making great strides. So the round robin is coming up. We're actually, Kelly Coleman from Medtronic is heading that effort. And we have controls, sample controls that are showing a response in irritation. And we are looking at doing some round-robin testing to validate the procedure for extractions. And then after we get the round-robin done, then we'll submit that to regulatory agencies, and we're comparing that to the animal test to show sensitivity comparisons. And all that data will be then submitted to regulatory agencies, and the ISO body to be able to look at what the in vitro offers. It's hard to give you a specific timeline, but this is something that's a very hot topic for a lot of labs and medical manufacturers out there. So lots of time and resources have been put into this test. So I do not see it being, at least if everything works out well as far as the comparison results in the round-robin testing, that should not be that much longer. We're looking at maybe in a couple years to be able to have reliable, replaceable in vitro data for in vitro irritation. And then sensitization would be the next one to approach. But right now, honestly, the chemistry can help evaluate a lot of animal endpoints, such as carcinogenicity, chronic toxicity, and even in some cases, like we talked about, systemic toxicity. So those are in vitro approaches right now that we can apply.
Allison, I have a question for you. Is direct mark UDI required for single-use implants, that is, devices not intended to be used more than once? And then the questioner specifies intended for use or reprocessing repeatedly as part of an instrument case or set until consumed, such as spinal implants contained within the instrument set.
Sure. So as the draft document is currently laid out, FDA does not, or that is one of the exceptions. So they're saying that the direct marking requirements will not apply to devices that the manufacturer intends to be for only one use or for use in a single patient during a single procedure, even if the device itself is processed or has processed multiple times before the single use on that patient.
Okay, next question. Let's say a manufacturer uses this question is a little bit. One second, I'll move on to the next one. If the FDA is moving away from the checkbox method, why is there an updated table for the same purpose in the update? Is there any concern with companies skipping to the new table to be more consistent with the previous revision? I'll turn that one over to Thor.
Yeah, so there's a couple of questions there. The first one is if the FDA is moving away from the checkbox method, why is there an update table for the same purpose in the update? Well, the new, and granted we haven't seen the final version yet, but the new approach seems to be more focused on, that's not a checkbox as far as testing goes. It's a checkbox for potential evaluations for endpoints. For example, you should evaluate sensitization. Now, historically you would just run a Magnuson-Caligman test or guinea pig max test to evaluate sensitization, but it's not necessarily telling you evaluate sensitization by testing. What it's telling you is that that's one endpoint we have to evaluate in the assessment. And so you go through the assessment evaluating systemic toxicity or genotoxicity, whatever endpoints are there. That may be testing, but it may be other endpoints or other ways to evaluate that endpoint. So the chart itself isn't necessarily a way to evaluate that. And then as far as I think the second part of that question was, what if they move to a new? How does it meet with the old standard, or has it compared to the old standard? Really, because we moved in the ISO document, we moved that chart into the appendix. What we're really trying to do is diminish the role of that chart in absolute, right? So once again, we really want to understand the device, evaluate the potential harm to the patient, risk to the patient, and then evaluate that risk to submit to the FDA.
Thank you. For long-degrading bioabsorbable materials such as PCL with a 3-year plus degradation timeline, how do you determine the length of implantation studies? It isn't reasonable to do an animal test out to 4 years, so how do you make a decision? Thor?
Another good question. So the standard has a term in it called steady state. So we want to get to a steady state of that implant material, and in some cases that steady state may be long. So what we tend to do, one is you got to remember we're using an animal model here too. So metabolism can be different depending on the animal used, and so the steady state may be different. So we try to do different endpoints. We try to get an idea of when the majority of the material has been absorbed. So you might have to do different time points within the implant structure. So you might have to do a 2 or 4 week or an 8, 13, 26 week implant to try to get an idea of maybe what's happening over time. So instead of just waiting to the very end of that timeline to see after the device is absorbed, we would prefer to actually get an idea of the story of the body happening over time. So that's usually the approach we use for a long-term implant or degrading implant.
Thank you, Thor. Next question, how do you know if they're leachable substances?
You have to test for them. So, all the biocompatibility tests, technically not all of them, but most of them look for leachables, which are just chemical compounds that come off under more clinical conditions. There are chemistry tests that evaluate leachables. So we will extract in a polar and non-polar solvent. We will run it through some chemical techniques like GCMS and an ICP-MS and HPLC and other machines, or basically what you're doing is looking for compounds that will come off that device. So you're identifying, quantifying individual compounds that are considered either extractables or leachables. And at that point, then you evaluate toxicity. So you can either evaluate the leachables either in a generic way, which is the historical way, which is just putting it into an animal and seeing what happens, or you can do a more individual plan where you actually identify and quantify those compounds and then look at the individual risks, either or the mixture risks, which are both associated in the ISO 10993-17 standard.
So Allison, I have a question for you. Why were only guidance documents from 85, 95, and 05 added to the C list? What about other guidances that are outdated?
Sure. So FDA put that together because they decided that, I think it was a public workshop last year. They said, hey, we should review the relevance of the old guidance documents and see if any should be thrown out or updated. So FDA has added them this year. The C list is sort of added this year. And FDA hopes that every year until 2025, they'll do a retrospective review. So next year, they'll look at 86, 96, and 2006 and ask for commentary on those.
One more question. What are the exceptions for UDI direct marking you referred to in your talk, Allison?
Sure. So I'll refer them to the draft guidance for UDI in section D. There are 4 exceptions. So any type of direct marking that would interfere with safety or effectiveness, like I mentioned. Number 2 is if the device can't be directly marked because it's not technologically feasible. Some devices are really, really small and hard to mark or etch. You can't attach a plaque to a dental screw. Third would be if the device is single use, like I mentioned earlier. And 4, if the device has already been previously marked.
Okay, thank you. Next question. Should tests be adjusted to the intended use? Like MEM elution cytotoxicity has time parameters that are far away from the intended use of certain products, such as surgical products. Thor?
Yeah, that's another great question. So once again, it's the difference between checkbox and scientific approach. Now, we would like to stick to the protocol as much as possible, which is usually a 24-hour extraction, which I'm guessing that is the question. But cytotoxicity can be a very sensitive test. So if that 24-hour extraction does not, if the test does not come out favorable, then one of the evaluations you can perform is to do a more clinically relevant extraction time. Let's say if it's only an hour contact with the body, then we can do an hour extraction and see what impact they may have. Or we can do like a 5x worst case where it's like a 5-hour exposure and see, or extraction, and see what kind of impact the cytotox may have there. So I wouldn't jump right to that approach. If you're going to do a cytotox test, it's usually better just to stick with a 24-hour. But if you know you have materials that are going to have issues after a 24-hour extraction, or if you do have issues after a 24-hour extraction, then that's one great way to kind of problem solve. Once again, it's all about scientific approach and addressing the risks versus benefit of the device.
Okay, I think we're running out of time. I have one more question for Allison. What is the current FDA guidance or thought for biocompatibility testing on reprocessed devices?
Sure. So Thor and I were just talking about this earlier today. So you can use biocompatibility testing sort of as a screening method for, to see if there are any residuals or anything left on the device. Thor, do you want to add on that as well?
Yeah, it's just a lot of the reprocessing has potential to manipulate the materials, which we talked about life cycles, understanding the life cycles. But also, most of these reprocessing detergents or chemicals are pretty toxic in themselves. And so if you are using these to reprocess and you aren't removing them all, then you have potential biocompatibility issues. So some of the testing that's used to help screen for these potentials, we talked about cytotoxicity. Most of the chemicals are extremely the cytotoxin is very extremely sensitive to those chemicals, so it's a great way to kind of screen potential residuals of a reprocessing chemical. Chemistry too, we can look at some residual testing. We can look at FTIR if it's a plastic on a material and compare it to one that's not reprocessed to see what kind of impact it's maybe having. So there's a lot of, we can do a gravimetric which looks at non-volatiles coming off, and then we can do an FTIR on the gravimetrics. So there's a lot of ways we can evaluate cheap and quick screening tests. We can look to evaluate the impact of reprocessing on a material.
Yeah, and I think when I was at the agency, a lot of companies would submit cytotox. That's what I would see the most. So yeah, I think having the ISO 10993 draft or the standard come out will be really helpful to figure out how to use the testing for reprocessing.
Okay, thanks guys. That's all the time that we have for questions right now. QMed appreciates your time and effort to make this webinar a success. Lastly, we want to thank everyone in the audience. We appreciate your attention and participation. We hope what you heard and saw was interesting and helpful. This presentation will be available shortly in an on-demand format. As a registered user, you'll receive an email with detailed information on how you can access the on-demand replay of this webinar. Please feel free to invite your colleagues and peers who may have not been available to listen to the event. This webinar is copyright 2015 by QMed. The presentation materials are owned by or copyrighted by Nelson Laboratories, Inc. The individual speakers are solely responsible for their content and opinions. Thanks for joining us and have a great day.